Now, I thought that with our brief survey of the human body that it would be a shame not to spend some time on the cells that built the Golden Gate Bridge, that built the pyramids, that built the Palm Pilot, if you want to take an extensive view of what these little masses of protoplasm can do. So I'm going to very briefly cover the two main cells in your cerebral cortex that are responsible for all of your behavior. So there are two types that are most common. These are cerebral, cortical cells. Two most common will be the pyramidal cells. You understand why I took my choice of words as to what we're respecting in society? The pyramids, pyramidal. And stellate cells. The pyramidal are the most common. And as the name implies, they're pyramid-shaped. And the stellate, as the name implies, they're star-shaped. When you get pictures of them from the slides, they don't always have a whole cell. You get parts, but roughly looking something like that with a short axon. The pyramidal cell has a long axon. So long axon neurons are classified as Golgi type 1. You'll see that in the literature. Golgi type 1 with a long axon. In contrast, stellate cells are Golgi type 2 with short axons. In fact, sometimes these are called local circuit neurons because they have such short axons carrying on functions within the local circuit. But what I want to elaborate a little bit to show you the dynamics of dendrites, we'll talk about the dendrites on the pyramidal cell. And first, we'll have the apical dendrite. So it means that it's just coming from the apex of the pyramid. Coming up here, this is my apical dendrite. Coming from the apical dendrite, we'll have oblique dendrites. Oblique dendrites. And these will be coming off obliquely from the apical. Somewhat like this, so we'll just put 2 for our oblique dendrites. And 3 then are the basal dendrites. Basal dendrites. Coming from the base of the triangle or the pyramid, they'll have first a primary, then it'll branch to a secondary, tertiary, and so forth, and can keep on going for many branches. So these represent basal dendrites. Maybe distinguish them from the soma by making this solid. But it's interesting because each set of dendrites receives a different input. So the apical dendrites are receiving subcortical axons. The oblique dendrites will be receiving from the opposite hemisphere, the contralateral hemisphere. Opposite or contralateral hemisphere. And the basal dendrites will receive intracortical input. Intracortical. What's the term for the same side? Have you heard it? Ipsilateral. So this would be the ipsilateral side. Intracortical on ipsilateral, which is same side. So when one is measuring dendrites and looking for changes with the environment, it's fascinating when you find only one set of dendrites is responding. We found that the basal dendrites in our work were responding most, and that was most exciting because the intracortical play is of the highest kind. So it gives you an idea of why one studies these dendrites. Now, one type of cell, which is called a Bet cell, it's the largest neuron. We gave you two examples of the largest. We gave you an anterior horn cell. So an anterior horn cell and the Bet cell are the largest, let's put them together, largest neurons. Where do we find the Bet cell? It's a pyramidal cell in area 4, layer 5. Bet cell, pyramidal cell in layer 5, area 4. So can you see it? Can you see where your area 4 is in your brain? And you know you've got 6 layers to the cerebral cortex, and you'll see these big ones. So when you see them, you won't think that this is due to some abnormality. They are normal. There are few of them. They inhibit anti-gravity muscles. Just to give you a little bit about them, we'll stop there, but it lets you know they have a special function. So that's just essentially all we have time for to give you the brief introduction of two types of cells. Now we want to spend the rest of the time dealing with the eye, this phenomenal structure that's designed to bring in light, to stimulate nerve cells, to give you images of the outside world. How many of your cranial nerves are related to eye functions? Half of your cranial nerves, 6 out of 12, are related to the eye. So, twofold purpose in giving these. So the first one then will be the second cranial nerve. What's its name? Optic nerve. So we know its function is vision. We have this, what's the third cranial nerve? Oculomotor. So it's going to the levator palpebrae to raise your eyelid, if you remember. and 4 of the 6 extraocular eye muscles. So we'll have a superior rectus, we'll have an inferior rectus, and we'll have a medial rectus, all supplied by the third, as well as the inferior oblique. Then we have the 4th cranial nerve. What's its name? Come on, what's its name? Thank you. What's it do? It's a pulling muscle. Good for you. So you remember trochlea, remembers a pulley. But where is it going? What's this muscle is this? What one's left out? Superior oblique, right. Process of elimination, you can learn by that, if nothing else. Superior oblique. And then we get 5. What's 5 doing for you? What's the name of 5? Trigeminal. What's it doing for your eye? What's it doing for the face? What's it doing for your nasal cavity and your oral cavity? It's sensory, right. It's sensory. You get a piece of dust on your cornea. That's 5th nerve. So it's sensory to the eye. We'll put cornea because that's what causes us most of our problems, getting something in our eye and it hurts and it itches. 6th nerve, what's it called? Abducens. Remember what it does? What muscle's it going to? Lateral rectus, right. And let's see, we've got 2, 4, 6. We need one more. 7. What's 7 doing for your eye? You've opened your eye. How are you going to close it? Circular. Every time you blink. What is it? Orbicularis Oculi. See why I review and review? It's up there somewhere, but you don't know how to retrieve it. And that's what we've got to train you on how to retrieve. All right, so this is facial. And it was going to your orbicularis oculi. So you can close your eye. Now, I think there's just one statement in here that I think is important, that I read that 70% of the receptors in your body are in your eye. Just to make the dynamics of this. Here we all kinds of receptors throughout your skin, but with the receptors in your eye, it said 70% were in the eye. What part of your brainstem does your eye develop from? You think it's going to be hindbrain? No. Midbrain? No. What's left? Forebrain. Good. We can work up to it. Sounds elementary, but when you don't know it, we've got to review. So yes, it's going to come from the diencephalon. The more you remember your embryology, the more you'll remember your neurology of the brain. So let's see. We've got a cross-section of the diencephalon. We have our central canal. And we have at the floor of the diencephalon, the migration of nerve cells that will form what's called the optic cup. Two eyes. So the optic cup has two parts. We have the pigmented retina, and we have the neural retina. Pigmented is the outer portion. And the neural portion, the inner. The pigmented layer prevents the diffusion of light to adjacent rods and cones. Prevents diffusion of light to adjacent rods and cones. So let's now develop our neural retina briefly. Spend a whole semester on it. Just introduce the basics. And this will be anterior. This will be our neural retina. Probably had this basic biology. We'll start with the light is coming through, comes all the way through the retina to the lowest level of cells. And the first layer will be the rods and cones. And these are your photoreceptors. How many rods do you have? About 100 million. How many cones? About 7 million. Why do you need rods? What do they do for you? Pardon? Night vision, dim vision, right? Night vision. Or dim vision. Why do you have cones? Acute vision. And color vision. How do you remember which is which? Cones begins with a C, acute, a C, and color, a C. All right? Just because frequently it makes it easier for you. Now the next layer of cells will be the horizontal cells. And what are horizontal cells doing for you? Lateral integration. So I have to make room here. Horizontal cells to lateral integration. And then we have bipolar cells. And they're carrying the impulse on up to the next layer, which is 4. So amacrine cells. What do amacrine cells do? They're again lateral integration. What makes them different? What's amacrine mean? They have no axon. A without. And we're working our way up to the last layer. What is it? The ganglion cells, right. And their axons now will form the optic nerve. Your second cranial nerve. But how do they do that, and what happens, and why does the light have to come all the way through all these layers to go out eventually? Why don't we just have the rods and cones catch it first, and then the ganglion cells come out? Good question. We'll wait for our evolutionists to tell us. But the axons from these cells now are going to collectively come out and go back to the brain. Giving an example. And as they pass through, then, the retina, we call the area the optic disc. What's another term for the optic disc? The blind spot. Because you can see there are no photoreceptors here. It's just neural tissue passing through, nerve fibers. So this is our blind spot. If we just look with your ophthalmoscope at your retina, when you get your eye exam, what is the optometrist seeing? This is retina. From looking into the eye and we'll see in the center of the retina an area called the fovea centralis. Fovea centralis. The fovea centralis has only cones. So we'll have the most acute vision. This is where you're focusing the light back into a specific area. So most acute vision is in the fovea centralis. And surrounding the fovea centralis, what do we have? An area called, it's yellow on the outside and white on the in. Looks like we have no more yellow chalk. This one looks yellow inside. In yellow, then, we've put in the Macula Lutea. Macula Lutea. Which means yellow spot. Macula is spot. Lutea is yellow. So it surrounds, so you know where your fovea centralis is because of the presence of the macula lutea. But what I'm leading up to is where is the blind spot in relationship to the fovea centralis. And so if this is my nasal side and this is my temporal side, so you can picture this on yourself, we'll find the blind spot about here. It's on the nasal side. Equals your optic disc or blind spot. Have you ever tested yours to be sure you have one? Anybody not tested it? You tested yours? Put your finger in front of your eye, in front of your right eye, and then look straight forward, and then move your finger to the right and see where your finger disappears. Do you see where it No, you're not looking, you're following your finger. That's not. Look straight ahead. Look straight ahead at your red shirt back there. And now I'll move my finger. And it disappears right about here. I don't see the finger, but it's crossing your blind spot. Can you get it? Some can and some can't. Try it on your own, but at least you know. And why do we teach this? How often have you pulled out from a stop sign, you've looked once, and you don't see any cars, and you pull out, and all of a sudden, where'd that car come from? That car was in your blind spot. So I always say, when you pull off from a stop sign, turn your head twice so you can catch it. But you'll find it's really very important for a safety factor. All right, so we've got a brief introduction to our retina. And let me be sure I've got all I want to give here. Yes, so let's just follow our optic nerve back so you get the general idea of how it's organized going to the brain. And somebody's sleepy. All right, let's take this. We can have lots of these coming back. All right, what have we done? Well, we have our temporal field of vision out here, our nasal field, our temporal field of vision. And we formed here what? The optic nerve. So if we cut the optic nerve, what are we going to see clinically? I've only cut it here. This is my right eye. This is my left eye. What are you going to see? Sure. Very simple. You've cut all the pathway that's coming from the eye. So if I cut it, you get right eye blindness. Isn't that simple? Nothing complex there. Let's put a pituitary tumor in. You've seen where the pituitary is. You know it's in the sella turcica, right? Part of your which bone? Pardon? I hear mumbles, but which bone? Sphenoid, sure. Terribly important. You forgot all that already? My, we'll have to put some of those questions in the exam, get you reviewing. So here we've got a pituitary. And the pituitary has a tumor. So, if a tumor, what are we going to see in our visual field? We're catching information coming in from the temporal field, right? So we're catching, these fibers are cut off. So you'll have in your eye no temporal vision. You'll have blindness out here in the field. So it's a bitemporal. We'll just call it blindness, but it's called hemianopsia, but I won't give you the word. We'll just put blindness to make it easier for you. But to figure out when you damage the visual system, you have to know what the pattern is so you can see the problems. This then is my optic nerve where it's crossing here. This is the optic chiasm. Optic chiasm. This is the optic tract. You get different problems with vision in peripheral vision or external vision if you have damage in various parts of the pathway. So this is my optic tract. And what's important, just so you've heard of it, is this loop here. These fibers, this area is my thalamus here, where they're having a synapse in the thalamus, all sensory pathways, synapse in the thalamus. We have the same nerve all the way from the retina, all the way back to the thalamus, and then we get new fibers coming to the visual cortex. But why do I mention this? Because this loop, called Meyer's loop, is very important anatomically to know that some of these visual fibers are going forward here instead of backward. Why is that important? When the surgeon does a temporal lobectomy, person has incurable epilepsy, and they want to get rid of the source, and it's in the temporal lobe. They remove the temporal lobe. But if they're not careful, see the temporal lobe's out here like this, and they'll cut it off, and they'll cut that Meyer's loop. So why it's important to know so-called deviance from what you'd anticipate. It's obviously a developmental thing. When the brain was growing, it carried these fibers forward and then they determined to come on back again. But that's Meyer's loop. And then back to your visual cortex. What number did we give to your primary visual cortex? It's V1, right? But, pardon? Area 17, sure. This is area 17. So many people work on the visual cortex, and I want you to at least be aware of what they call it, or V1. So do you appreciate everything you see now, what it has to go through? And this is just minuscule compared to what really goes on. So now, let's see if we've given you the basics here. Let's look at the coats of the eye, because we've got to protect this retina. So the coats, we're going to have three. One will be the retina. Two will be the uvea. And three will be the sclera and the cornea. So we've done the retina. Let's get our eyeball. We need a big area to work. So this is our optic nerve. This is our retina. And so we can start putting on our coats. Let's put on the uvea. It consists of three parts: the choroid, the ciliary body, there, and processes, and the iris. So we're going to put in our choroid coming around outside our retina. So this will be choroid. And the choroid is primarily vascular. And then it expands into this ciliary body. Choroid was number 1. This is number 2. And it has processes, little finger-like projections. And what are those little finger-like projections holding? What do we need here? Pardon? A lens, sure. So they're going to be holding the lens. What's a lens made of? Have you ever dissected an eye? Have you ever taken the lens out and put it on a newspaper, and what happens to the newspaper print? Magnifies. When you go out into the classroom, the kids love it because a lens is made of epithelial cells, and yet they can magnify. Hold it in your hand. They change their chemistry. So we haven't had that before in the body. We'll continue. We'll come back if we have time, because we want to finish our coats. So the other structure that we need will be the iris, and the iris is going to come around here. So this is as we see the iris in this position. If we take the iris and look at it from in front, we'll see just the disc with the pupil. And here would be your pupil here. And we'll have the muscles that we've mentioned before in the iris. We have those that are circular. What were they doing to the pupil? These are circular muscles. What are they going to do to the pupil? Constrict, right. Constrict pupil. And then we have the radial muscles going from inside out. These are radial muscles. What's going to happen when they constrict? Dilate, sure, you can just see if these are muscles, they do this, pupil enlarge. So dilate. So you don't just have to memorize, you can figure them out. It's so nice when students come to the lab, the office hours, and they're beginning to work with it, the information, and they can figure things out. It's just beautiful to see. Dilate pupil. All right, we have one more coat to go. We have the sclera. So sclera will be made of dense, they're white, collagenous fibers. So what's the common name for this layer? When you look at somebody, they've been studying all night. Your sclera is supposed to be white and it's red because you've worked so hard. So the sclera, the white of the eyes, will be connective tissue. Protecting, it's like the dura around the CNS and PNS. So this is our sclera. And anteriorly, it's going to become translucent. And what's it going to form? The cornea. Again, the cornea is made of collagenous fibers, but they've changed their chemistry so that light can get through. So these are collagenous fibers. But they're translucent. Anybody had a corneal transplant or know of anybody who can? Wonderful things they can do today, right? Now, at the junction between the cornea and the sclera, junction of cornea and sclera, two more things I'd like to say, and then we'll show slides. junction of cornea and sclera are what are called the canals of Schlemm. Another one of these scientists with a great name. Canals of Schlemm. There is fluid posterior in the chambers in here that's being formed, and it has to get back into the venous system. So it gets back into the venous system through the canals of Schlemm. So fluid posterior to cornea. Does anybody know what you call that fluid? Aqueous humor. It goes into canals of Schlemm back into the venous system. So what happens if those canals of Schlemm are blocked? You block the flow. You increase the pressure. What did we call the condition? Glaucoma. You've heard of glaucoma. It's when canals of Schlemm are blocked. So if we block canals of Schlemm, you get glaucoma. So go in with a laser, open them up, and get some drainage, get rid of it. What do we do when you get a cloudy lens? What do we call a cloudy lens? Cataract. What do you do when you get those today? You get a lens transplant. And the best place in the world for you to get a lens transplant is this Turner down in San Leandro. The head of neurosurgery at UCSF once said the best gift he could give me was to tell me the name of Tom Turner in San Leandro. And everybody I've known, I've said, go down to him to get your cataracts removed and get your lens replaced, and they don't have problems. Other places, they're still having problems. I've never given, but since he gave it to me, I figured I'd give it to you. We've got a few slides. Let's go through. All right, I just wanted you to see the histology of the cortex to know that this is filled with pyramidal cells and stellate cells, but layer 1 of the cortex has no pyramidal cells or no stellate cells. So you always know where layer 2 begins, because 1 has no pyramidal. It has other cells, but not pyramidal or stellates. Next one. And this is one of your pyramidal cells I've shown you. The reason it's so blurry is because it's cut thick so that we can get the branches in here. Cut at 150 mu, and we normally work at 10 mu or 6 mu. Here's the big apical dendrite. Don't get many other dendrites coming off, and the axon's so thin you don't see them. But we know these are not axons, any of them, because they have dendritic spines. Next one. This is a stellate cell. Stellate cell. It's got its processes. One looks a little bit thicker here, but on the whole, it doesn't have a pyramidal-shaped body. In the next one. And these are just the eye muscles. What fill your orbit, then, will be obviously your eyeball, your eye muscles, but in addition, it's packed with fat to cushion the eyeball. In the next one. And this gives you the basics. You can see the artery that's coming into the retina is from what major artery coming into the skull? No, the internal carotid. Do you remember the first branch of your internal carotid was the ophthalmic artery? When you look at an eyeball through an ophthalmoscope, you'll see the artery that's come up through the optic nerve all over. It's just wonderful to look at somebody's ophthalmic artery so clearly. But then you can see that we'd have the retina and then the pigmented epithelium showing here. There are different names that I didn't give you, but you come up into where you'll have the ciliary body, the ciliary processes and ligaments that suspend the lens, and it's the contraction of muscles in the ciliary body which allow the lens to change shape. And next time I'll tell you how that works. But here's the iris here. Here's the pupil between the two ends of the iris. And this is this anterior chamber that's filled with the liquid. And the canals of Schlemm are here at the junction between the sclera and the cornea. Right there. There are other parts. We'll have to give a little bit more on Monday. Next one. And this gives you a picture of the retina with the rods and cones and a horizontal cell for lateral excitation. And then the bipolar cells, which are synapsing with the rods and cones and with the ganglion cells. And here the amacrine cells, which are again lateral distribution. And then your ganglion cells connect with the lateral inhibition cells and the bipolar cells. And here are your ganglion cells whose axons will make up the optic nerve. In the next one, and this shows the optic nerve, the optic chiasm. Here's the pituitary. So you can see pituitary tumors can encroach. When they remove a pituitary, they keep you awake so you can tell whether they're touching your optic chiasm to see whether you get any diminution of vision while they're trying to take out the tumor. Optic tract coming back to the lateral geniculate of the thalamus, and then it will loop around and come back to your cortex. It's a beautiful dissection. These are your olfactory tracts and olfactory bulbs very nicely here. Next one. That's it. Enjoy your holiday.